So it's a great pleasure to have Briony Yorke to come and give the school seminar today. Briony originally did an MChem in the chemistry school at University of Leeds before continuing at Leeds in the Asbury Centre for a PhD, working with Arwin Pearson and Robin Owen at Diamond Light Source. She then moved to the Centre for Ultrafast Imaging in Hamburg to complete the PhD. And while she was there, achieved a prestigious Sir Henry Wellcome postdoc fellowship to work on time-resolved crystallography. She moved back to the UK in 2020 to start her own research group at University of Bradford and was recently awarded the Early Career Prize for biological structures from the British Crystallographic Association. And really looking forward to hearing your talk, Briony, and over to you.
Okay, great. So let me attempt to get everything going. So let's share this. So can everyone see my slides just before I start? Great stuff. So today I'm here to talk about the work that I've done on time-resolved crystallography, or in a more interesting way, perhaps making molecular movies, because that's with time-resolved crystallography, that's really what we're trying to do. But I thought I'd start with an introduction basically on what it is as a structural biologist that I'm interested in. So for structural biologists, really, we're interested in finding out what biological molecules look like. So what is their structure and how does that determine their function? So how is the structure, the three-dimensional ordering of all the atoms in a biological model? How does that determine how it works? But there's one thing that often we miss out from this question, and really we shouldn't just be trying to look from a static structure and trying to relate that to function. There's a missing piece, and that piece is dynamics. How does the structure move? How does it reorder itself to lower the activation barrier of a chemical reaction? And that's really what we're now trying to find out using X-ray crystallography. So first of all, the structure. Using X-ray crystallography, we can get very, very detailed models of what proteins actually look like. So here we've got an old style three-dimensional model of a protein. And we used to have to build them with sticks and balls on these massive grids to actually see what they looked like. But now with all the developments and advances we have with computation, we can build these models in the computer, we can rotate them, we can move them around and see really what it is they're made up of. And we can see the structure of the active site in enzymes. And that's really important because it shows us how ligands might bind into those active sites and how the reaction might take place. We can get clues on the chemical mechanism for the reaction using just the structure. But really, when we come to function, we want to know what it does. So we need to know how it works. And this can really help us then with medical developments and other things. If somebody has a disease based on an enzyme not working properly in their body, why has it gone wrong? Is there perhaps a mutation in a specific amino acid that is essential to function? Can I make it do something else? Now, this is a really interesting question for biotechnology because enzymes are perfectly designed to catalyze chemical reactions. But what if we want to catalyze a slightly different chemical reaction that isn't naturally found in nature? Can we change the enzyme? Can we edit it? Can we make it make something else for us? And then finally, can I make it work better? If somebody has an enzyme that's, again, not functioning properly, can we try to change it somehow and determine how we could make it better? Can we use drugs that would somehow alter the structure or the function of an enzyme? So these are all questions that as structural biologists we're interested in, but static structures aren't always enough to help us answer these questions. So then we come to dynamics, and my favorite quote about crystallography, from a sort of biological perspective, that's no, crystals don't wriggle, and if it doesn't wriggle, it's not biology. really is this wriggling, it's this movement, it's the dynamics that really tell us how something behaves, how it lives, or how it works and functions. So then we have to decide, in our crystalline system, are the dynamics we see in any way related to biology? A crystal is the very, very unnatural form of a protein or an enzyme. Can they even work in the crystal? Will they even turn over? Are the static structures that we have even related to how they look in a cell? Well, here we see something that's really interesting, and that's the concentration of proteins that we have in crystals is very, very similar to the concentrations of proteins in the cell. So the cell is absolutely jam-packed with different types of proteins, all undergoing their different functions. A crystal is made up of the same protein, but the concentration is very similar. But what we see here, you can see it even in the GIF, is a protein, an enzyme turning over in the crystalline form. We can see from this color change that it's actually doing its job. So there's one key caveat here. If there are no large conformational changes during catalysis, many proteins remain crystalline during turnover. So as long as we don't have huge changes in the structure that could push against the crystal contacts, then we can see the whole catalytic process happening in the crystal. If there are large structural changes, what's likely to happen is once we initiate the reaction, the crystal will crack, it'll fall apart, or it'll just become slightly more disordered, and we'll lose any X-ray diffraction data that we could get from that crystal. So here we can see this reaction happening, and it's turning over. The green color is disappearing. We're going from quinone to quinol, and the reaction that we can see there happening quite happily in the crystal. So how do we see these dynamics? So this is probably one of the most famous examples of time-resolved crystallography. So here we can see this is the first ever motion picture made. So in 1878, Edward Muybridge wanted to film a galloping horse, and he wanted to understand how they move, how they run, and try to improve this. So there was a lot of money in breeding horses back in those times. So there was a huge push to try to improve the gait of horses and try and identify problems with it. But to do that, they had to understand how horses run. And they wanted to find out whether a horse's feet are all ever off the ground at the same time. And the only way they could do that was to understand the dynamics of how a horse runs. You can see a photograph of a horse and you can assume that its legs probably help it to move, but without seeing the videos of how it runs, it's very difficult to get that detailed analysis of it. So macromolecules are dynamic and flexible objects, but when we do crystallography, we take an ensemble measurement, and that sees all the conformations at once. So all these structural changes that are happening in our horse, they get blurred together when we do a traditional X-ray crystallography experiment.
So for 1 second, Briony. So for some reason it's gone to presenter view, so it's not quite as easy for us to see the...
Sorry.
Yeah, that's better. Thank you.
Clearer. So yeah, when we do X-ray crystallography, what we're doing is we're blurring together all these dynamics, and we actually lose the information. The legs just become a blur and we can't see anything. So we need a time-resolved experiment, something that can capture the horse in each of its different states and tell us how those states join together through time. So that's just what I said. And then so here we have the blurry horse. It's very difficult to determine anything about the way that it runs. And this is the problem we have in a traditional X-ray crystallography experiment. We see very clearly the parts that don't move, but anything that's moving during our experiment is blurred out and lost. So again, here we have the first time resolved experiment. We put all of our static images together that were taken at different time points, and we can really get an in-depth analysis of how this system is changing through time. And we can actually see that, yes, at some point during a horse's gallop, its legs are all off the ground at the same time. So now we're using this idea of a time-resolved experiment with X-ray crystallography to apply this to proteins and enzymes. So what's really important for us now is to make sure that the experiments we do are on the correct timescale. So biological processes happen across a huge range of timescales. From the things that control the very fast chemical reactions, such as electronic motions and bond vibrations, they happen on the femtosecond timescale. So that's 10 to the minus 15 seconds. And then we get down to the very slowest enzymatic step, which can take minutes or hours. So what we need to do is we need to make sure that we can match the time resolutions that we can measure to what it is that we want to see. So we have a number of techniques available to us as X-ray crystallographers to do time-resolved experiments. We can use something called an X-ray free-electron laser. This is an extremely powerful X-ray source, and this really lets us access these very, very fast timescales. We can watch photosynthetic light-activated reactions happen over femtoseconds. We then have a very, very specialist technique called Lowy crystallography. It's quite a difficult technique to do, but for a long time, for 20 years or so, we've been able to watch these picosecond to nanosecond reactions happening using Lowy crystallography. But the main thing that's held us back with using this technique is the availability of the equipment to do it and problems with data processing. So what I'm focusing on, and I know Mike does work with synchrotrons as well, is synchrotron radiation. Now, in the UK, we have a synchrotron X-ray source, and it's very, very widely available. Anyone can use this source. It's free at point of access. So it's something that I wanted to look at and see what we could see on these slower timescales that would be of interest to people, but also, can we change the experiment somehow to try and get into these faster timescales and make it easier for people to be able to do these experiments and have them as part of the sort of general structural biology toolkit rather than just a separate method that only the sort of very, very specialized crystallographers use. So here are our X-ray sources. We have this synchrotron, and here we have on the right-hand side a free electron laser. So this synchrotron is diamond light source in the UK, and its intensity really is what determines the time resolutions that are available to us. So a synchrotron can produce about 1 × 10 to the 12 photons per second. And this gives us time resolutions of microseconds to hours. Whereas the free electron lasers, which there are none in the UK at the moment, are able to give 1 × 10 to the 16 photons per second. And what the difference in this intensity means is we can get much, much faster resolution. Here we can get to femtoseconds. But as you can imagine, a free electron laser is not cheap. This picture here is the one where I used to work. It spans 2 states in Germany. So it starts out in the state of Hamburg here in Daisy, and then 3.5 kilometers later is where we actually collect the data and do our experiments, and that's in a different state called Schönefeld. So I'm just having to move my slide, the camera bit around so I can see my whole slides. Okay, so how do we do a time-resolved experiment? Well, generally, since about the 1940s, when George Porter and his friends came up with these experiments, we've used a very, very specific technique that hasn't really changed much. We use something called pump probe experiments. And what this means is at time 0, we trigger a reaction. And we usually start the reaction using a laser. And the reason why we want to use a laser is because it allows us to start the reaction in all the molecules very, very quickly. But as you can imagine, that means we're limited to reactions that are light sensitive. So in a light sensitive system, we can initiate a reaction in all the molecules in our sample in a femtosecond time frame using a femtosecond laser pulse. If we were to say just mix together an enzyme with its ligand, this would take a long time. It would mean that we're waiting for the diffusion to happen into the enzyme. And that means that all of our molecules are then reacting at different time points, and we get that blurring that I mentioned before. So with a pump probe experiment, what's really important is we have a way that we can trigger our reactions very, very quickly. So after we trigger the reaction, we wait a particular time delay, and then we use our probe. So our probe is anything that can look at the system. In the very, very earliest experiment, the probe would have been a laser, so we could do spectroscopic measurements very, very quickly. But what we do with time-resolved crystallography, the probe is now the X-ray beam. So we measure our diffraction over a particular time frame, and that's what gives us our time resolution. So the amount of time it takes to record a measurement from the X-ray diffraction is our time resolution. So what we have to do to get a full sort of molecular movie to see the full turnover of the enzyme or protein is we have to change this value of delta T and take diffraction images at each different time point along our reaction coordinate. And we have to build this up many, many times. So we have to repeat the same measurement multiple times usually. And that's because if we use a very, very short probe pulse, we don't get very strong diffraction on our detector. The strength of the diffraction is determined by the intensity of this pulse. So if the intensity is low, we have to measure for a longer amount of time, and that really limits our time resolution. And then we have to repeat the experiment at these different time delays to build up a series of time-resolved data sets. But the limitations here are, as I said before, we have low signal to noise, and we only get one data point per cycle. So for an irreversible reaction, we need lots and lots of samples. In a reversible system, it's quite easy to do. What we do is we excite the system, measure it, and then we allow it to relax back to its starting state and repeat. But for most enzymatic reactions, that's not feasible. And for biological crystals, one major problem is radiation damage. The thing we're using to measure our structure destroys it at the same time. I'll talk a little bit more about that later. So one way that we've come up with to deal with this problem of irreversibility is serial crystallography. So serial crystallography is very different to a traditional X-ray crystallography experiment where you have to grow one beautiful, perfect crystal, put it into the X-ray beam, measure it as many times as you can before it's destroyed by the X-rays and get to your structure. What we do now is we use hundreds and thousands of very, very small crystals. They're about 20 microns in diameter. We mount those in these solid supports that are like a mesh. So we just pipette them onto the mesh, remove the liquid, and very, very quickly, we have mechanical stages that scan through the crystals into the X-ray beam and allow us to collect the diffraction. And then what we do is we have to add all these images together. Hopefully the crystals are in a random orientation and we've managed to get all of our different measurements. And then we can add them together, scale them, and come up with a structure. So this would have been impossible about 20 years ago. The computational power needed to do these experiments has just reached the point where it's feasible. And so now we're really able to utilize things like pattern recognition and artificial intelligence in order to do these experiments and process the data. So I think from a free electron laser, one data set could easily run into the sort of terabyte size. So we're having to deal with these huge amounts of data, hundreds and thousands of images. So I just want to show you something that this is really what made me want to get into time-resolved crystallography. This is the video that really excited me. So here we've got myoglobin bound to carbon monoxide. And this is a reaction that we could initiate with a laser. And these measurements were taken using Lowy crystallography all the way back in 2003. And it was an extremely challenging experiment. But I saw this presented in about 2011 at the American Crystallography Association. I was so excited, I went home and told my supervisor that this is what I wanted to do. So we see here, we've got the heme group of the myoglobin, and we have the carbon monoxide is bound above it. And what we're looking at here is changes in the structure. So the white areas are the atoms that don't move. The color magenta shows where the things were before they moved, and the color green shows where the things are moving to. So if we focus on the carbon monoxide, we can see that it must come unbound after the initiation, after the laser pulse. It's moving above the heme group and slightly to the right of that. Then we can try to follow it around the structure. It starts appearing there on the right below the heme group and moves around before eventually it rebinds in this system. So I was just so excited that we could actually film chemical reactions happening. So at this point, we were applying for a time-resolved beamline at the synchrotron in the UK. We wanted this Lowy beamline so we could do these complex experiments in order to get time-resolved information about our system. But unfortunately, we were unsuccessful. We were left with monochromatic radiation. And we were in a position there where we kind of had to think, well, how can we do these experiments with the equipment that we have available? Oh, okay. So I just want to talk a little bit about reaction initiation. So I mentioned before that biological processes span a very wide range of timescales, but the majority aren't light sensitive. Unfortunately, we can't just initiate a reaction using a laser like we did here with the myoglobin. So how do we do these experiments on our enzymes? Well, for very, very slow reactions, we can use rapid mixing techniques. Here we have microfluidics that force our enzyme crystals and their ligand together and cause very fast mixing to occur. But really, for anything faster than milliseconds, what we need is a laser initiation. So there are a number of ways to do this. We can use a temperature jump. So this uses an infrared laser to input a lot of energy into the system and initiate a reaction. We can use photoisomerization or direct photocleavage. So this is what would happen in, say, photosystems 1 and 2, inherently light sensitive proteins. But for things where they're not inherently light sensitive, your average enzyme needs something called a photocage. So here at the bottom, if you imagine we have a ligand and it's got something attached, a big group attached to it that prevents the reaction from starting. We can soak that into our crystals so it goes into the active site of the enzyme in its inactive form. When we then photolyze the bond between the photocage and our ligand, the photo cage comes off, and our ligand can then turn over. So that's really how we're going to initiate the reactions in our crystals. There's a number of ways to do this. So we can cage the substrate. That's quite easy. We've done that a lot. The results aren't necessarily easy to look at, but the actual process of creating these photo cage ligands is quite easy. We can cage the active site. This is quite interesting because we can add photo cages to the actual enzyme rather than the ligand. And this makes it very, very widely applicable. So if we can photo cage an amino acid in the enzyme, we can photo cage any enzymatic system. Although it's, again, not always straightforward. It can change how the protein folds. It might interfere with its natural dynamics, with how it's actually supposed to work. So that's something, again, we'd like to avoid caging the active site specifically. Or we can try to allosterically cage the system. So what that means is we put a photo cage outside the active site, and we try to make it so the cage holds the enzyme in an inactive structure. So the active site is clear, it can do its job properly. As soon as the photo cage releases, the enzyme goes into its natural conformation and the reaction starts. So this is something that we'd like to work on more, but we have, again, a number of things that we need in order to do these experiments. So a photo cage has to have clean and efficient photochemistry. It has to have a good quantum yield. We don't want to only be partially activating the system. It has to have adequate absorption at wavelengths longer than 300 nanometers. So proteins and enzymes absorb at 280 nanometers. If all of our laser light is being absorbed by the protein, that's going to affect the initiation. So we want the wavelength of our photoactive groups to be outside of that range. It has to have good aqueous solubility, which is actually really challenging just to get it into the crystals in the first place. And we need to generate inert photo products. So if we do our photo initiation and the products that are released after and go around and affect our active site and our reaction, that's no good. It has to be clean. The photo products have to not interact with the system once they've been produced. And then the release rate of the free substance has to be faster than the process observed. So our photo cages are time limited again. And we have very basic nitrobenzyl groups that work on millisecond timescales. These coumaryl groups are much more interesting because they work on the nanosecond timescales, and then we can try to alter these in order to change where they absorb and how efficient they are. So that's what my colleagues over in Hamburg are working on at the moment. So the challenges for our time-resolved experiment, well, we have photo initiation. We also have radiation damage. So we've got ionizing radiation that we're putting into our system. And another issue is that not all the changes that we want to see in our reaction are structural. So these two groups would look very, very similar in an X-ray crystallography experiment. So we need to be able to observe these changes as well at the same time as changes in the structure. So radiation damage, I mentioned before, that's the fact that our probe, our X-rays actually damage the system as we're looking at it. We are altering the system as we observe it. And just for comparison, 3 gray, that's joules per kilogram, that's a measure of our X-ray dose, is enough to kill a hamster. We regularly introduce about 30 million gray into our crystals during an experiment. So we're way, way above the sort of biological safe doses. And what happens when the crystals are exposed to these X-rays, when all that energy is dumped into the crystals, is they explode. So here you can see some virus crystals in an X-ray beam. and we can go through and shoot them, and we could possibly get one useful diffraction image here before the system is so damaged that anything we see is just a blur. And so here we have some numbers, how radiation damage changes the structure as we collect data. So as we said, normally, as we do an X-ray crystallography experiment, we're working on this mega gray dose scale. So even at the down in the gray, in just one gray, we're starting to produce photoelectrons. And you can imagine the damage these free electrons can do to the system and how they might change the chemical reactions that are happening in our system. So photoelectrons appear almost instantly. We then start to see localized damage. So here, this is showing a disulfide bond that has been broken by the X-rays as we've done our experiment. And then as we progress with the experiment, we start to see global damage, and that's the entire crystal structure starts to fall apart, and we lose the intensity of the diffraction that we see. So even if we've managed to collect a data set, we see diffraction, what we need to take into consideration is that way before we start to see a reduction in our diffraction intensity, structural changes have occurred, and the electrons, the photoelectrons, have flooded the system. So here's just an example. This is the X-ray reduction of myoglobin. We can see after 7.5 seconds of exposure, this is very, very old data, so the timescales are much slower than what we look at now. But after 7.5 seconds of X-ray exposure, we've seen that the myoglobin has almost been completely reduced. This band here is the Sorey band here. The first point at about 426 nanometers. We then see shifts in the Q band, and also at 600 nanometers, we can see this formation and then dispersal of the solvated electrons. So what we need to do is we need to monitor the radiation damage, and also look at these changes that we have, these electronic changes which might not necessarily show up in the system. And to do that, we use online spectroscopy. That means when we do our X-ray experiment, we have our crystal in the beam. The X-rays are coming towards us in this picture, and the spectroscopy system is above and below our sample. So from the same sample at the same time during the same experiment, we're collecting both diffraction and spectroscopic information about our system, and this is incredibly useful. We can see these electronic changes. We can follow the formation of sulfated electrons. If it's something with a chromophore in it, we can watch the radiation damage occur like we have done in myoglobin. If you look coming into the sample in this direction, we have our cryostream. So normally during a general static X-ray experiment, the sample's held at 100 Kelvin. And what this does is it slows down the radiation damage. It slows down the diffusion of the photoelectrons, so they can't go around our protein causing changes. But unfortunately, this isn't appropriate for time-resolved crystallography because it also freezes out these functional motions of our protein. So that's something that we can't use to help us prevent radiation damage now. So we have to very carefully monitor that as we do our experiments. So new methods. How do we increase the time resolution of synchrotron experiments? This is really what my research is focused on. So as I said before, we have the synchrotron, very widely available. This is in the UK. We've got 7 MX beamlines that are quite easy to access, I think. But again, it's very slow. You can get up to microseconds if you're lucky. If you want to use a free electron laser, that's much more challenging. We have no beamlines in the UK for this. There are 3 in Europe. It's a very new technology. It's a very specialized technique. And hopefully one day it will become a standard experiment, but at the moment it is just not feasible. We cannot all do every single enzyme experiment on a free electron laser at the moment it has to really be set aside for things that are extremely important, like photosynthesis for solar energy, and also experiments that are quite easy to do a synchrotron to then translate to the free electron laser. At the moment, I don't believe there's been many studies involving photo cages, for example, at free electron lasers. They're too challenging at the moment to take the risk of using that big piece of kit just to watch something that might not work. So how do we increase the time resolution of our synchrotron? Well, you remember before when I introduced the pump probe experiment and said that the time resolution was really limited to the probe pulse. So how long do we need to measure to get a useful signal on our detector, which again is determined by the intensity of the X-rays that we have. Some of the things that stand in the way of time resolution is the timing between the pump and the probe, but this is something that's electronically controlled now. It's very easy. If you're using an optical delay line, which you may have heard of if you're a spectroscoper, there's something that might be a bit more challenging, but at the moment, this doesn't cause us any restrictions in our time resolution. And again, the pump pulse width. So how long do we need to illuminate our crystals with light in order to initiate the reaction? So again, that's determined by the photo cage or our system, the state of our system. So what really I was trying to focus in is how can we get around the probe pulse width? How can we get around the intensity of our X-ray beam and those limitations that we have? So how do we increase the signal to noise ratio that's limited by the intensity of our X-rays? Well, we can increase the exposure time, so that's we have a longer probe pulse width, but this reduces time resolution. We can use a higher flux, so we use a free electron laser or a Lowy experiment, which uses polychromatic X-rays rather than monochromatic X-rays. Or we have to average together multiple measurements. So that's we have to repeat the experiment over and over and over again to build up signal on our detector to get a measurable thing. But again, that's difficult with an irreversible enzymatic reaction. And then there's a few technical things we can do. We can reduce the background scatter. We can use bigger crystals, but this isn't feasible for photo initiation. Because the laser is absorbed as it passes through the crystal, our size limit really for laser initiation is on the order of about 50 microns. So bigger crystals diffract better, but we can't use bigger crystals. We can reduce the detector noise. So developing detectors has been a huge push in X-ray crystallography, and we basically have noise-free detectors now. These photon counting detectors that we use are very, very fast and have very, very small signal to noise ratio. But it still doesn't help us get to these higher time resolutions. To do that, we can do something called multiplexing the data points. So instead of repeating the experiment at the same time point over and over again, we can record multiple time points in a single image. So we're essentially increasing the exposure time by adding together multiple time delays. So this is called multiplexing with the Hadamard transform. So what it does is it allows us to split a probe into multiple pulses, and each one is measured and integrated on the detector. So each of these thin slices are added together onto the detector and read out as a single image. And then we can back transform the data using the Hadamard transform to get out our individual images. And I'll explain how that's done now. So Hadamard time resolved crystallography is based on the Hadamard transform. And the Hadamard transform basically tells us how we can group together measurements to improve the signal to noise ratio, but still be able to do a mathematical transform to get our initial results back out. So If we have 3 different objects with 3 different masses, and we want to measure their mass with scale, but every time we use the scale, there's an error associated with that measurement, an uncertainty. We could either weigh each one multiple times and take an average, but actually the best way to do it that gives us the best possible answer, the lowest uncertainty, is to measure these items in groups. So if we measure the apple and the ball together, and then the ball and the car together, and then the apple and the car together, and we record these results in a vector, what we then do is we multiply that vector by the inverse of the matrix that told us which ones we needed to weigh together. So the matrix tells us how we group together our measurements. And then by multiplying that by our results by the inverse of that matrix, we get out our individual masses for each item with a lower error. And that's basically because we've taken fewer measurements than measuring everything separately. So we know we've managed to measure the apple there twice, but because we were measuring 2 things together, we were grouping them together, we lower the uncertainty. So it's just a way of averaging that gives us something called a Fell-Getz advantage. And that just means we can improve our measurements without averaging, but by using these groupings that are determined by the matrix. So how does this work in crystallography? We used a pulsed beam to record a series of time points per image. So each diffraction image contains the information from all of the time points that are given by each row of the matrix. So the sequence that we have these pulses is determined by this matrix. So for n time points, we still have to repeat the experiment n times, but we're not having to take the same single measurement over and over again, which means it doesn't matter if the systems are reversible. We just measure the full reaction from separate crystals. So, okay. There's a number of challenges that are associated with this, obviously. And the first one that we came up again was, how do we pulse the X-ray beam to allow us to measure these groups of time points? Well, the first way that we did it, and the easiest way to do that, is just to switch the detector on and off, but keep the information from the previous measurement, integrate it together, and then read out at the end of each measurement run. So our detectors here, this is a Pilatus 6M. It's a little bit outdated now, but it's a very good detector. So a single pulse is about 100 microseconds, and the rise fall time, so that's the slope on the pulses at either end, is about 10 nanoseconds. So this was perfect for our early experiments, and we can now happily collect data on the 100 microsecond time scale using this method. Another way to do it is to use a very fast chopper. So if we use this detector gating, our crystal is being exposed to X-rays continuously. We're only measuring the diffraction at each particular time delay. So we're introducing a lot of radiation damage. There's a very high dose that's happening whilst we're not measuring anything. And this is kind of a waste. We want to be able to turn the X-rays on and off, really, not just the detector. So one way to do this is to use a chopper. This is a spinning shutter, so it blocks out the X-rays except when there's this little gap, and that allows us to pulse the X-ray beam. So a prototype that I've made, here's the results. We can see a single pulse is about 40 microseconds. the rise fall time is about 10 microseconds, which is a bit high. It adds a bit of uncertainty in our measurement. So it's not great. And actually, it turns out that trying to design a chopper that can work on the sort of nanosecond timescales is an extremely difficult engineering challenge. So this is something that has an advantage in that the X-rays the crystals aren't being exposed to the X-rays whilst we're not measuring anything. But the disadvantage is the engineering challenges to make something safe that spins that quickly are extremely difficult. But again, here's the prototype that I made of a Hadamard shutter. So you can see on the inside, we have just some evenly spaced circles there for timing. Our Hadamard sequence is in the squares in the middle row. So that turns the X-rays, or in this case, the laser, on and off according to our Hadamard sequence. And again, on the outside, we just have regularly spaced timing gaps for our control lasers that kind of feed back into the motor and ensure that the speed remains constant. So we managed to get a total measurement time of about 1 millisecond. So that's the full rotation of the chopper. And each time point was about 4 microseconds. And we measured that the signal to noise ratio was approximately 8 times better than a conventional measurement. So we managed to collect 4 microsecond time resolved data in an experiment that would usually take 10 milliseconds. So we've managed to get quite a good improvement in the time resolution using this method, but for anything faster, it just seems to be unfeasible now. Oh, and one thing that's interesting about the S matrices is they rotate. So if we go back to the S matrix, you'll see we just move one position to the left for each row. And that's incredibly useful for making a chopper because it just means we don't need a new disk for every row of the matrix. We just start one position later. So that was quite interesting, but it's not something that I think is going to pan out in the long term. We need better ways to do that. So again, serial crystallography, we can look at irreversible reactions with photocage ligands. We look at one pulse sequence per crystal at a fixed orientation. Again, 100,000 microcrystals are needed and they're in solid supports. And the time resolution for these experiments at the moment is determined by the gating of the detector to produce the sequences and the bunch structure of the synchrotron. So that's how the electrons are going around the synchrotron, how flat the intensity of X-rays is that are coming through the synchrotron. So the very first experiments that I did using this method were just proof of principle experiments. Can we see changes of structure using Hadamard transformed data? And we looked at radiation damage in thaumatin crystals. So what we were expecting to see is these yellow atoms here are sulfurs. So these are disulfide bonds in thaumatin. And what we wanted to see is using the transformed data, can we see those break with increasing X-ray dose? Okay, so I'll explain what we're seeing here. The green meshes show where the atoms move to, and the red meshes show where they move from. So in the original structure we had before we didn't input a huge dose, we had these nice disulfide bonds. But as the dose increases, we can see that they swing out to the sides, they pull apart, and the disulfide bonds are broken. And we see that in both our control data and our Hatrix data. Now, I had a hard time persuading my PhD supervisor that this experiment would work. Would we be able to do this mathematical transform and see the same thing that see what we were expecting to see in the crystal, or were we just going to get a bunch of nonsense numbers? And I think she was quite excited when she saw these results, that we can actually use this method to watch disulfide bonds breaking. So these are just the disulfides that we see falling apart in thaumatin. And that was really the proof of principle to show that the maths worked. So then we wanted to move on to a system that had all the limitations that we've discussed before. So it's an enzyme, aspartate decarboxylase. Its substrate is not photoactive. Its substrate is just aspartate. And what happens is the enzyme takes aspartate, takes off a carboxyl group, and we get beta alanine. That's what the enzyme does. It's part of the pantothenate biosynthesis. So we photocaged our ligand using this big coumaryl group. And then we initiated the reaction, and what we wanted to see was the change from aspartate to beta alanine in the active site and all the structural changes that accompany that. It's not been so straightforward. Working with photocages is very difficult. As you can imagine, they're not the natural substrate for the enzyme, so their binding efficiency is quite weak, which means we're looking for very, very weak intensity of this part. No, I don't know why this graph always spins round, but it does. We followed the reaction initiation using our online spectroscopy, and we were quite happy to see that we had peaks that were associated with the photocage. So the photocage had definitely gone into the crystal. So at that's where it absorbs around 350 nanometers. And we can see when we switch the X-rays on, the photocage starts to fall apart. So this was our experiment. We used a 7x7 S matrix that normally, ideally, we're going to be using much larger matrices, but we have to show the experiment works first. The bigger the matrix, the longer the experiment takes, but also the better the improvement in signal to noise. So for now, we're still working on these proof of principle smaller experiments. So we just have some technical information there about the experiment, but what's really interesting here is that we're integrating 1 millisecond frames, which at that point in time, we would normally get no information out, but we were integrating 7 of those, which helped us to see some diffraction on our detector. So we collected data from 20 chips, and in the end, we ended up with 50,000 integrated images. And that was about 7,000 per Hadamard sequence. So each sequence had 7,000 images associated with it, which was just enough to try and get a structure. And so here we see the preliminary results of our time-resolved experiment using photocages and the Hadamard transform. I'll explain what you're seeing here. The pyrovoyl group in aspartate decarboxylase is where L-aspartate binds, and that is then turned over to remove the carboxyl group. So here we can see that before the pump, before we initiated the reaction using our laser, we see something that fills the purple mesh bound to our pyrovoyl group. After 4 milliseconds after that initiation, whatever it is definitely moves. And I know this is not very exciting if you're not a crystallographer, but to me, this was amazing. We've actually managed to see some structural changes associated with a photocage in an enzyme active site using the Hadamard method. So again, it's not a massive change. We're still processing data. We're still coming up with better ways of collecting data and getting complete data. So this data wasn't ideal, but we saw something, so we decided to carry on. But the amount of complexity in this experiment made it very, very difficult for us to be able to make any sort of concrete conclusions. So I've moved now onto human gamma D crystalline. Now, this is an incredibly interesting protein. It makes up your eye lens and it's expressed in the fetus. Once the eye lens is formed, the cell machinery dies. And this is one of the very, very few proteins in your body that is never renewed. Once your eye lens is formed, that crystalline has to stay soluble and folded for your entire life. It has to be exposed to a huge amount of UV radiation and all kinds of other environmental factors, but it remains folded and soluble until at some point in our lives, these proteins start to degrade and clump together, forming cataracts. So we know that there is a UV element to the formation of cataracts. So somehow the UV destabilizes these proteins after a certain amount of time, and they start to unfold and clump together. So here we can see parts of the crystalline protein. And at the bottom there, we have a sulfur group. So we've got a cysteine here at the bottom. Now, in our dark structure, before we initiate the experiment, there's something very big attached to our cysteine group. Okay, and then after intense UV radiation, we see that that large group disappears. Now, we know that the eye lens is also full of reducing agents like glutathione. Glutathione fits quite nicely into this density. And we can see after UV initiation, the glutathione comes off and is no longer bound. So what I'm proposing is that in our eye lens, we have a certain amount of these reducing agents, which can bind to the disulfide, to the cysteine groups and prevent intermolecular disulfides forming, which clump the proteins together. After a certain amount of UV radiation has been absorbed by the eye, these glutathione groups degrade and so can no longer perform their protective role in the eye lens. And we've seen actually in clinical studies that the concentration of glutathione decreases over time, and it's incredibly low in people who have cataracts formed. So this is something now that I want to go on and try to prove by doing more experiments. But really, this has shown how we've taken time-resolved crystallography and applied it to a medical problem and shown the mechanism that causes the formation of cataracts, or at least come up with more clues that will help us to get to that mechanism and perhaps come up with ways to treat cataracts, to reverse their formation, or prevent them forming in the first place. So currently, we can do these hat-trick experiments at Diamond Light Source in the UK and at the T-Rex beamline at Petra 3 in Hamburg. And we have all the software tools available to process the data. Thankfully, the data processing now is very, very run-of-the-mill compared to the free electron data processing. All the work that's gone into free electron laser experiments has really helped to boost our research. So the challenge now, the thing that's really holding us back is reaction initiation. Very few systems are inherently light sensitive, so we need to work on these photo cages. So I didn't do this by myself by any means. I had a lot of help. So obviously my work was done at the synchrotrons in Europe, mostly at Petra 3 in Hamburg with Tom Schneider and Gleb Borenkov. Diamond Light Source in the UK with Robin Owen and Graham Winter. Robin was actually one of my PhD supervisors. And then at the ESRF with David von Stetten, who now works in Hamburg at the Petra 3 beamline. And then at the University of Hamburg, my colleagues Arwen Pearson and Niels Hughes were very, very much involved in this research. Anka Pusha was my master's student whilst I was in Hamburg, and she actually engineered the photo chopper, the rotating shutter that I showed you earlier. Joe Doyle and Marta Sands work on the photo cages. Sam Horrell and Michael Agg that actually built the time-resolved beamline in Hamburg, and so were incredibly helpful and productive during that time. And then I moved to the University of Leeds for a short period and had support from these people. And I now work at the University of Bradford, and I'm just trying to get my feet to get these experiments going again. So thank you for your time, and I'm sorry I think I went on a bit too long.
Okay, thanks very much, Briony. Fantastic talk.
Oh,
Really inspirational stuff. Do we have any questions from the audience? Either in the chat or by unmuting yourself?
I've got a quick question. So hopefully quick anyway. I'm a bit obsessed with eye lenses.
Oh, great.
Because we use them to look at basically in fish, at least. I'm not sure about other taxa. That's what I wanted to ask you about. Was in fish you get these kind of like layers, like tiny little onions, and you can peel them back, and in the inner layers we can look at what they were eating, you know, kind of isotopically when they were juveniles, and so we can kind of retrace their kind of lifelong dietary needs and that kind of stuff. So yeah. I'm curious, do you know why those layers formed based on what the work that you're doing? And do you know also if fish eye lenses are similar to all other taxa, out of interest?
I don't. I'm currently working with the School of Optometry on a grant proposal, and my collaborator's role is to talk about the specifics of the eye lens, because it is very interesting. I think, is it zebrafish are widely used for this, for the eye lenses. But yeah, I'm not sure if the structure of the human eye lens is in layers. But again, it's quite interesting for a crystallographer to look at these proteins because of the ordering and the organization involved in actually making the lens is related to crystallography. One really interesting thing I found is that certain forms of cataracts will diffract if you put them in the X-ray beam. There is a type of crystalline cataract that can actually diffract, which is what drew me to them in the first place.
Oh, super. Can I just send you a load of eyeballs and you just like do your magic with them and tell me more?
Well, we really want to actually start working on actual eye lenses. So I'll be in touch, I think.
I'll email you. Okay, thanks.
Definitely.
Fantastic. So I have a sort of future-looking question, which is there's lots of upgrade projects going on for the synchrotrons around the world. How fast do you think it might be possible to go in the next few years if you combine the multiplexing approach with the new beamlines that are coming?
With the upgrade to Y24, I think we'll be able to get to, if we use specific ways to pulse the beam, we should be able to get to picoseconds. So the way that I see pulsing the beam is bouncing the electrons into a different orbit. So if you've got all your electrons going around the synchrotron in one orbit, you can bounce them to a higher orbit according to the Hadamard sequence, just using magnets. And then that means the electrons are causing the pulses, which means the X-rays pulses further down the line are very, very nice, very clean. So that's ideally, that's the next upgrade I want for Diamond.
That's going to be amazing. Yeah, any more questions?
Yeah.
Hi, Mike.
I just wonder what the chopper is made of that you chops the X-rays, because that must be taking a real beating and must be scattering X-rays all over the chopper as well. So do they last very long or what?
No, generally not. We actually did a Lowy experiment once, diamond light source. So that's where you use multiple wavelengths X-rays. So it's much more intense. And we used an aluminium chopper and we melted it within about 5 minutes.
Yeah.
So if you're going to use anything heavier, that means that spinning it really fast is exceptionally dangerous. So it's become something that we've sort of pushed aside now. It was after we spoke to the engineers, they were just like, it's never going to happen.
Are there any other metals that you could try apart from aluminium? I mean, I don't know.
I mean, anything, but the heavier it is, the better it withstands the X-rays.
Yeah, I suppose so, yeah.
You could have a lead chopper, but it would be so...
Something with a high melting point as well, I guess.
Exactly, yeah. You could have a sort of crystal chopper. So something that diffracts the X-rays into a different path. But another method would be to have a mirror made out of a crystal that you can move, or you put an electric current over it and it changes the crystal lattice. So it changes the direction that all the X-rays are being diffracted in as you put current through it. But the problem with that is the amount of X-rays that actually get diffracted onto the new path, it's not 100% efficient. So you're losing intensity, and the whole point is we'd want more intensity.
Oh, yeah.
It's trying to balance those needs.
It's a tricky problem.
Any more questions?
Is it possible to put glutathione back into a lens?
Yeah, this is the idea that I've had. So I was a bit cheeky before. What I showed you with the crystals are grown with DTT. So in my expressed protein, there was no glutathione there that I could identify the origin of. So I think that's DTT acting in the same way that glutathione would act in biology. So now what I need to do is go back, do the experiments again, grow the crystals with glutathione, check that it's the same, and then see if we can look at developing ways to get glutathione back into the eye lens. My big idea, my 20-year idea is doped contact lenses, where you have a controlled release of these therapeutic agents that stop cataracts forming. But
a UV sensitive photo cage, it does the thing. then you're safe.
Yeah, exactly. Just take exactly, yeah, that's so true.
Great. Good.
Brilliant. So unless there are any more questions, so we'll thank Brian again for a fantastic talk.
Very good. Fantastic. Yeah, very good.
Oh, thank you. Thank you.
Very good.
Thank you for inviting me as well.
Very good.
Yeah, really interesting. Thank you.
Brilliant. And we'll see people at the next seminar.
Yeah. Bye-bye and thanks.
Thanks, everyone.
Bye, thank you. And please
Bye-bye.
feel free to get in touch with me regarding research. I'm always looking for new
My lens in the post.
Yeah, thank you.
Bye-bye.